Hi everyone, I'm gonna get started because I just heard the ding, but thanks for coming to my talk today. My name's Dan. I'm a PhD student at University of California, Berkeley, and today I'm gonna be talking about some of the preliminary results for my first pilot season in the field this past winter working with some salamanders. So as we all know, environmental change is going to cause a lot of organisms to respond in novel ways. And for ectothermic species that I study, like salamanders, this is going to require them to maintain some sort of thermoregulation or water balance efforts. So what we have here are some salamanders in their ideal habitat, indicated by this blue circle. And environmental shifts imposed by things like temperature or altered precipitation regimes are going to require these salamanders to respond, and usually respond in evolutionary ways. So understanding these responses is really the core of what I'm interested in for my PhD research. So these big questions I'm interested in are how exactly organisms will respond, and whether or not we can make models, predictions of their vulnerability, but also if we can sort of map their future distributions based on this contemporary variation. So now I'm just going to run through some common responses that organisms might have. So I think most people think of dispersal as the most common mechanism. So what we have here, the salamanders are dispersing to a more suitable habitat that might match their original niche space. But for the species I study, and for many species many of us might study, they're dispersal limited. So the idea of being able to move tons of kilometers, it's not really possible for the species I study. Some other mechanisms is persistence through phenotypic plasticity. So this is essentially expanding the range of phenotypes that organisms or organisms within the population have, and sort of these expanded phenotypes allow them to persist. Another mechanism is behavioral thermoregulation. So what you can see here is on the left, there's like a salamander hidden under a tree. So it's hiding under this shade of this tree in a cooler like microhabitat in this larger, warmer macrohabitat, essentially. But importantly, both of these mechanisms sort of don't really get at ideas of local adaptation or ideas of standing variation and how that could enable persistence. So what I'm interested in is sort of characterizing local adaptation and standing variation, and particularly because this could be a more effective mechanism for dispersal limited organisms and ones that sort of have to, like, quote unquote, adapt in place. But of course, this assumes populations in the present have the necessary phenotypic and genetic variation to enable adaptation in the future. So characterizing local adaptation, like I said, can sort of be difficult, and sort of teasing it apart from phenotypic plasticity can be difficult. But it's critically important because thermal trait variation is what allows organisms to persist and what will allow organisms to persist. So for a lot of this talk, I'm going to be exploring some of this characterization of local adaptation, really getting at the interplay between environmental variation and phenotypic/thermal trait variation, and how those two are correlated, and what that can tell us about the persistence of a species across its distribution or a portion of its distribution. So the system I'm studying this in is the Ensatina salamander within California. So they form a number of different subspecies, I'll call them for this talk, across their distribution. And they're most well known for being a case of like ring species within California. But another aspect that they haven't been explored as much in this species is its sort of response to environmental variation across its range. So California has a really great latitudinal diversity in terms of an environmental gradient from north to south, but also in terms of elevation. So there's a lot of differences in temperature in vegetation regimes across the Ensatina range in California, and specific subspecies are restricted to entire groups of these particular environmental regimes. And on an organismal level, they're a useful system to start studying these questions because they are dispersal limited. They show marked phenotypic and genetic divergence, like I just mentioned. And there's also phenological considerations. And what I mean by that is These salamanders, like most salamanders, they're pretty inactive during the summer. They will sort of be burrowed in the ground in a state of inactivity. But then they're most active in the winter during the wet, rainy season. So there's a phenological component as well. So that leads me to this main question I'm interested in, which is whether or not environmental variation is correlated with potentially adaptive thermal trait variation, which I'll describe in a second, in local populations of Ensatina salamanders in the Bay Area of California. So the two main phenotypes that I am interested in is these two here. So the first one is critical thermal maximum, which I'll call CT max for the rest of this talk. And that is essentially just the loss of writing response of an organism after heating it up for a steady amount of temperature per minute. And then the other trait is sort of looking at color, so really thinking about dorsal brightness or quote-unquote melanism and how that can interplay with thermal tolerance and also different environmental data. So I've talked about the two phenotypes that I measured. Now I'm going to talk a little bit about the environmental variables that I've collected. So I did this on both the micro and macro scale. So for microhabitat variables, I've listed them out here, but the important thing to take away is that most of them are related to either temperature, humidity, or some metric of vegetation cover or leaf litter composition. And that's really important because on the right here, you can see like individual, they're really small, like probably about like this big. So they are really tied to microhabitat conditions. So this individual was found like right under the log at the layer between the log and the soil. So they exist at these really small spatial scales. And I could not put photos of salamanders in the presentation just because they're so cute. So here's some examples of what they look like. So on the left, you can see here just how small they are and how like dispersal limited they might be. And you can also see here different subspecies as well. So on the left is the yellow-eyed Ensatina xanthoptica. On the right is the Origensis subspecies, which goes all the way up to British Columbia, all the way up the west coast. And here's just a photo of me collecting these microhabitat variables. So they really are at a small spatial scale. So the exact log or the exact rock where I found the salamander is where I'm recording these suites of variables. Moving forward to the macro scale, so a lot of these were taken from remote sensing data. So these are like about 250 to 500 meters in resolution. So the major two are vegetation cover, or NDVI, and also air temperature. But I also, throughout this project, developed new layers of soil temperature just because, as I mentioned before, these salamanders are in the leaf litter, and during the summer, they're burrowed in little like mammal holes or in small little crevices in the soil. So they're really tied to the soil. And we don't really, at the moment, have really fine scale layers of soil temperature, especially for California. So I decided to make them, essentially. So what I did for this little sub-project is I took layers of land surface temperature, NDVI, and albedo, and then I used the Meriflux tower network across California, which they have actual values of soil temperature for about 23 years. And then I took the two and put those in a regression model to be able to predict soil temperature at unsampled locations across the state of California at a pretty fine spatial scale. So this is about 500 meters, give or take. And what we can see here is that there's pretty good model support for this, for predicting at unsampled locations. And just on some preliminary analyses, when we try to validate estimate soil temperature with actual reference values of soil temperature, we also get a pretty tight correlation as well. So what you can see here is just like a zoom in of my study area, one, so that you can see that it is a pretty fine spatial scale, like sites are only a few kilometers apart. But more importantly, we can see that soil temperature also begins to vary pretty dramatically between different areas. So a lot of those yellow areas are sort of built up urban cities of the Bay Area, and then those cooler areas are either coastline or more forested. So this model performs pretty well, and it's something I'm sort of actively developing and hoping to release towards the end of this year. So if you or someone you know who's working in California might benefit from this, definitely get in touch with me during the conference. So just moving on to some preliminary results from this. So the first thing I found here is a correlation between individual length and the CT max, which is the upper temperature limit, like I mentioned before. And individual length in salamanders is sort of a good proxy for the age of an individual. So the main takeaway from here is that as individuals get longer or they get older, they have generally an increased CT max relative to their younger counterparts. But this is also might be expected a little bit just because we know from thermal physiology and thermal trait literature that body size generally tends to correlate with CT max. In terms of our other phenotypic trait, what we find is that as individuals get longer or older, they tend to get darker, so brightness decreases. So you could say that there's increased case of melanism, as opposed to younger individuals, which tend to be more brighter than older individuals. And we're able to visualize this as well, just looking at these like color-corrected images of individuals. You can see that younger individuals tend to be brighter, and older individuals tend to be darker as well. Now, if we try to correlate our two phenotypes together, so this is after correcting for body size, so using the residuals, we can see that there's not really a significant trend, but there does appear to be something where increases in brightness are tied to increases in CT max. So it could be that younger individuals are less reflective, so they might have higher CT max. But I think this is something that if we increase sampling size, it could start to tease apart this relationship better. Now, if we're looking at the environmental correlations to these two traits, the main thing we see with CT max is that there's a non-significant but slight trend where increases in evaporation rate at the site of capture are tied to increases in CT max, which makes sense because if an individual if there's an increased evaporation rate, there's probably an increase in temperature or thermal stress that's operating in that location. If we look at NDVI, so like a macro habitat variable, we find that increases in NDVI are actually lead to individuals being darker or is tied to individuals being darker. So it could be related to how individuals absorb or lose heat as well. But we can see NDVI does vary pretty dramatically across this landscape, so it could be tied to other things like crypsis of the organism and its environment. And finally, the last thing we see is that generally, as the standard deviation of the soil temperature increases, so the variability across years, we see that individuals tend to get darker, which could be related to the fact that darker individuals are also better related or have a higher capacity to thermoregulate, both in terms of being able to gain heat and dissipate heat more quickly. And this is just a map showing how the variability of soil temperature across the study area is pretty variable as well, just kind of similar to that soil temperature map I showed earlier. So in conclusion, we can say that there might be a sort of age-related or developmental correlate just because younger individuals tend to have reduced CT max. But the main takeaway is that thermal trait variation seems to be linked to vegetation cover, soil temperature variability, and water loss. But I think one of the main takeaways I have from this pilot data is perhaps humidity regimes might be a bigger factor for salamanders than temperature regimes. So exploring that in the future might be more useful. And also that darker individuals might be more well equipped to thermoregulate than lighter individuals. And that becomes apparent probably due to their association with like higher NDVI or more variable soil temperatures. So future directions that I want to take this in is definitely expanding across the whole range of Ensatina across California. So this photo is from Southern California. You can see here it's a dramatically different environment than the Ensatina and like the forested redwoods of Northern California. And the last thing I want to do is try to develop some desiccation or water loss experiments to see whether or not humidity regimes might be having a greater impact. And I think that's the end. So yeah, thank you.
Hi everyone, thank you very much for coming to my talk. My name is Antoine Perrier. I'm a postdoc at the University of Virginia in the Galloway lab. And today I'm going to show you a little bit of my research that I did there, where I got interested in reh population and particularly their distinct evolutionary histories, phenotypes, and adaptations. So to start off, you all probably know that species have often very well-defined distributions, but these distributions can change over time, and particularly changes are associated with changes in climate. For example, during last glacial maximum, about 20,000 years ago, the Earth was much cooler as it is today, and species were not found where you find them today. They were often constrained in what we call glacial refugia at low altitudes and latitudes. After last glacial maximum, as years got warmer and ice sheets started retreating, many species experienced what we call post-glacial range expansion, where they essentially colonized newly suitable habitats towards higher latitudes and altitudes. And in this context, we can define different evolutionary histories across the range. On one edge, we have the leading edge, which are young colonizing population at the front of the range expansion. And on the other side of the range, we have a re-edge population, which are often relicts of those that persisted in these glacial refugia. This means that re-edge populations are often the oldest from across the range. They often have a history of long-term persistence at the warmer range limit, and nowadays they're often found in isolation and occur in very distinct and marginal habitats. And all of these attributes make this population really interesting for me to study because that means they might be very different from the rest of the range, especially in their adaptations. And understanding these differences might be very important to consider when we think about how climate change will affect species distribution, because we often predict that re-edge population will go extinct under climate change because habitat will become less and less suitable. However, we are seeing more and more examples of species where this is not the case and re-edge population are doing better than what we expect. So how different are re-edge population? This is a question that has obsessed me for most of my postdoc, and I've decided to answer it or address it in Campanula americana. It's a North American plant species that is found across forests, across most of the eastern United States, as you can see on this map. And a little bit about its life cycle. It's a monocarpic plant which sets seeds in fall. Seeds will rapidly germinate, and then seedling will be exposed to winter, where they will get vernalized, and then in spring, plants will start bolting and then flowering again in late summer. In this species, I'm defining the re-edge as a lower latitudinal third of the distribution, or every population below 35-degree latitudes. I know it sounds very arbitrary, but I'm going to show you that it's not. One of the main reasons is that we think this part of the range was a likely glacial refugium. This is a projection of where habitats were suitable during last glacial maximum, and as you can see, at the re-edge is where this species could have persisted during last glacial maximum. So it's likely that population may have been persisting there until today. However, the rest of the range must have been colonized after last glacial maximum. During my postdoc, I also did some phylogenetic work, and I found that population at the re-edge or below the 35-degree latitudes are also very distinct from the rest of the range. You can see a phylogeny that are generated based on rat-seq data, with each branch representing a population and each color representing a different genetic cluster assignment. And the main thing I want you to retain is that most population north of this delimitation belong to a single phylogenetic lineage, indicating a single post-glacial range expansion event. But population at the re-edge belong to a very different phylogenetic lineage that has diverged before this post-glacial range expansion, and they also belong to different genetic cluster. So genetically really distinct. If we look at contemporary condition, we can also see that this delimitation at 35-degree latitudes represents a change in climate, where for most of the range we find population in temperate continental climate, but at the re-edge climates are more subtropical. And these differences in climate also translate in differences in habitat suitability. Here's a projection where habitats are suitable currently, and you can see that for most of the range, habitats are suitable, but suitability rapidly declines at the re-edge. And this is an indication that populations, they occur in habitats that are very different from the main niche of the species. These differences led me and my former undergrad also to look at whether differences in climate are associated with differences in phenotype, indicating diverging climate adaptation. And we looked at that by looking at differences in phenology, particularly linked to flowering time and vernalization. We first looked at whether flowering varies across the range based on observation natural population, and we found that while for most population across the range, flowering occurs later towards high latitudes, which was associated with a delayed start of the growing season. This was not the case at the re-edge, where the pattern was completely flipped, where population flowered later towards high latitudes. And because this was associated with a change in climate, it was the first good indication that there was some diverging climate adaptation happening there. We also looked at winter conditions, and we found that at the re-edge, winters are very mild, which is expected under subtropical climate. But it's also an indication that these populations are receiving almost no vernalization, which is weird if we think about the fact that this plant requires vernalization to flower. So we wanted to test whether population at the ridge have adapted to this unique condition by taking population from across the range and exposing them to different conditions of fertilization that differed in length, and then looked at how many plants flowered per population. And as you can see here, we found that for most of the range, if you reduce the length of winter, plants flower less, and in some cases don't flower at all. But this was not the case for the re-edge, where this population flowered very well, regardless of which conditions they were exposed to, even in the lowest vernalization treatment, indicating that they have adapted to these unique conditions. Okay, to summarize what I did so far, I found that population at the re-edge are likely glacial relicts. They're genetically very different from the rest of the range. They occur in very different habitats. They also show differences in phenology associated with this difference. So this led me to the main question I want to address today, which is whether all of these differences can also translate in differences in local adaptation. So I tested for local adaptation by performing a large common garden experiment where I raised 22 populations from across the range in 3 common gardens. One garden in Kentucky, if that works, in Kentucky represented the climate at the core of the distribution. The garden in red represented condition at the re-edge, and there was also an intermediary garden in South Carolina. For each of these gardens, I first raised seedlings of each of these populations in the laboratory, and then I had a first cohort where I transplanted seedlings in fall before winter to test for the effect of winter conditions. And I had a second cohort where I also raised seedlings but exposed them to controlled condition of vernalization and then transplanted them in spring to test for an effect of the growing season. And I had a total of about 4,000 individuals in this experiment. You can see what one of the gardens looks like at the beginning with the little seedlings that I transplanted on the right. And this is what the same garden looks like at the end of the experiment where all of the plants are flowering. And over the course of this experiment, I tracked several fitness estimates linked to survival, transition to bolting and flowering, but also how much each plant reproduced. And this allowed me to calculate for each population in each garden the lifetime fitness across most of the life cycle. The next thing I had to resolve was how different are the conditions that each population is adapted to compared to what they're experiencing in each common garden. To make it short, I calculated several environmental variables based on PRISM data for the 5 years before the experiment and then summarized them using a PCA, which you can see projected here. And I found that most of the environmental variation, unsurprisingly, was centered around the temperature axis, which explained most of the variation. And so this is the one I'm going to focus on today. I also calculated the same variables for each of the common garden for the length of the experiment based on data loggers and weather station data, and also projected them on this PCA. And this allowed me to compare how different are the environmental conditions that each population experienced at their site of origin compared to the condition in each site. And for this, I calculated for each population in each site delta PC1. And with this, I was able to test for local adaptation. And so essentially, I looked at, in a given garden, how does lifetime fitness of different population vary depending on how different is the climate of each population compared to the climate that they're experiencing. And in this context, if in a given garden a population has a delta PC1 of 0, it means it's a local population. Its climate at the site of origin matches the climate it is experiencing in the site. If for this population a delta PC1 is positive, it means it's a population coming from a colder environment, and vice versa if delta PC1 is negative. And of course, if there is local adaptation, I'm expecting local population to outperform foreign populations. Here's the result of the central garden. Each of these dots represents a population with on the y-axis average lifetime fitness, standardized within this site, and on the x-axis, of course, delta PC1. So the main thing I want you to see here is that local populations have higher fitness, so there is local adaptation. The pattern was a little bit stronger in the northern garden and was mainly driven by the fact that population coming from warmer environments had lower fitness, and this was mainly due to the fact that this population take much longer to flower. But in this site, growing season was way too short for them to complete entirely their reproductive cycle. Now for the site that was really interesting me, the re-edge, you can see that the pattern was really different. There was very strong local adaptation, with local population completely outperforming foreign population, which in some case had a fitness of 0. And this was mainly due to the fact that this population came from colder environments, had high vernalization requirement, but at the re-edge, winters are very low, or short, so they were just not receiving enough cues to flower. To exemplify this, here you have pictures of the Central Garden in Kentucky and the Ridge Garden in Florida during the bolting phase. And you can see that in the Northern Garden, where winters were long, all of the plants bolted and are getting ready to flower. But this was not the case for the Ridge, where only a fraction of them bolted. And I don't know if you can see it, but many plants stayed at the rosette stage, here circled in white. And these plants did not receive the vernalization cue, so just never flower and just die off without reproducing. Okay, so to summarize what I learned in this common garden experiment, I found that there's generally local adaptation across the range, but as I expected, it varies depending on range position. And particularly, it seems that ridge habitats are really marginal compared to the main niche of the species, as we found in the species distribution model. And this was mainly due to the fact that winters were too mild for most population to actually be able to reproduce. However, even if these habitats are very marginal, it seems that local population or ridge population were able to really well locally adapt to these conditions, thanks to their low vernalization requirements. With this, I'm arriving slowly towards the end of my talk, and I hope by now I have convinced you that ridge population are really cool to study because they are so different from the rest of the range. They have distinct evolutionary histories, they occur in very different climate compared to the rest of the range, and this can lead them to develop distinct phenotypes or different adaptations, and this can lead to strong differences in local adaptation. And as I mentioned in the beginning, it might be very important to consider these differences in adaptation when you try to predict species response to climate change, because it seems that in our case, ridge population are already adapted to warming winters, so at least for the winter part of climate change, they might be actually fine. And it might be a population at mid-latitude that are at greater risk of extinction because these ones still require vernalization, but under warming winters, they might just not be able to reproduce. And with this, I would like to acknowledge my funding institution, all of the sites where I did my common gardens, the help of all of the members of the Galloway Lab, and you all for your attention. And if you want to follow what I'm doing, here are my contact information. This is a QR code to my website. And I will take questions. Yes, in the back.
Thank you for such an interesting talk. Fascinating about the rear edge, so I'm sorry to ask about the central site, garden site. I just wondered if I could trust my eye. You did say you saw local adaptation there, but would it be fair to say that it was...
It was extremely weak, very weak, yeah. Which is very surprising that if you just go a few miles south, it starts really dropping, or like the local adaptation becomes much stronger because we are entering subtropical climate, which is really not what you would expect to find the species, except for the ridge. Other questions? Yeah.
Did you try any habits, and how would they be doing, like sort of migration or anything like that?
Sorry, can you repeat that?
Did you think to assess the fitness of any hybrids to maybe?
Oh, I have hybrids. I haven't raised them yet, so TBD. Yeah, we'll see. Yes.
Do you think it's possible the rear edge would colonize the mid-latitudes, or would that be the dominant?
That's a very good question. Maybe. But the thing is, they're starting to show signs of reproductive isolation with other populations, so maybe that might be detrimental, but we can talk more about it later. Thank you.
Hello, everyone. Thank you for coming today. My name is Lainey Galland. I'm a postdoc at the University of Arizona in Jesse Rick's lab. And surprise, this is not an adaptation talk because someone forgot to change the title of the talk, and here we are in an adaptation session. But I will say that we're going to talk about a really cool, really ancient, very large trout. Hopefully that's some sort of consolation for everyone. So we're going to talk about hierarchical genetic structure in the world's largest salmonid and second most ancient extant salmonid in the world, Hucho-Taimen. Oops. So a little bit about Hucho-Taimen. They're known as the river wolf. They're very voracious predators that attack at the surface in this big explosion of energy. They grow up to 2 meters in length and up to 50 kilograms. I argue they actually grow larger, but those specimens are rare. They're mainly piscivorous. They mostly are feeding on other grayling sister species Lennox, which are the most ancient extant species. And they were widely distributed across Siberia for quite some time. I think I'm skipping slides. There we go. They were originally widely spread across Siberia. You can see, if you're looking at this range, this map, which may be hard to see on the smaller screen, this range extends all the way north to the Arctic, south through southern China, all the way in the east to the Pacific Ocean, and to the west almost into Eastern Europe. So a very, very wide range. Now, these fish have been under a lot of different threats, and we've seen range declines across some of their range. These red sections represent some of the areas where the fish are actually extirpated. The Volga, the Ural, and the Pechora basins no longer can hold taimen. And the threat to taimen populations are really similar to those of all other cold water salmonids. Of course, we have climate change, which I haven't even opened the door to with this system, but also urbanization, pollution, lots and lots of sheep and goat mining, or grazing. Goat mining, it's a thing in Mongolia. Open pit mining, Mongolia is the number 1, 2, and 3 exporter of copper, gold, and silver in the world. And then overfishing and illegal fishing. And then, of course, hydroelectric projects, which are a serious concern in an area that is developing energy independence. So this has led to an estimated 30 to 50% decline across its range, and a listing is threatened by the IUCN. But these are all sort of blanket statements just to try to put some sort of protection into place for taimen because there's been so little known about them. This was a rush to try to protect taimen. So what do we know about genetic variation in taimen? Well, we largely lack studies that look at population genetic structure within river basins. Again, this map, the scale on the bottom, which is very difficult to see, is a 1,000-kilometer scale. So you're looking at the Arctic Ocean in the north, all the way through southern China in the south, again, the Pacific Ocean, and then pushing towards Eastern Europe. And I think you can see my mouse here. Nope, you can't. Okay. These are enormous river basins. So the entire red chunk there is 2 river basins. And you can see this is a study on just a few different markers here, mitochondrial DNA markers, and we get these monophyletic groups separating the large basins. And this makes sense given what we know about the natural history of taimen. They're very large individuals. They're capable of moving up to 100 kilometers, 150 kilometers based on tagging data that we have. And it would make sense that they might be really large panmictic populations. However, we don't really know if that's the case. It could be a case where we see finer scale genetic structure within populations. In this point, this map shows points where maybe one individual was captured and run through an analysis. So we really need to understand the consequences of habitat fragmentation as these hydroelectric projects are being pitched. So my objectives here were to look at differentiation among and within watersheds. So large scale, similar to what was done before, and smaller scale. Wanted to compare genetic diversity estimates across the range and estimate evolutionary history and how it underlies genetic variation. What could have happened in the past that might actually underlie what we see now? So how do we do this? How do we go capture taimen? We go fly fishing for them, which is by far the most awesome part of my job. So here we collected samples pretty opportunistically across northern Mongolia and in southeastern Russia. The purple you can see here I'll refer to as the Arctic drainage. This is a very, very large scale, so that's a 400-kilometer scale in the top panel, lower left of the top panel. And that purple region is the entire upstream area of Lake Baikal. So all the headwaters of Lake Baikal that flow north into Lake Baikal, the largest, deepest freshwater lake in the world. And then it eventually flows north to the Arctic Ocean. And then there are 2 Pacific drainages here. One is the very, very large green basin you see where samples were opportunistically taken in Mongolia in the upper watershed. The other is the Tagore River Basin, which is highlighted in the lower left panel. It's that red box in the top panel. The Tagore Basin scale is, that entire scale is 12 kilometers. So these populations are very, very highly proximate, ones that I didn't expect to even analyze separately. So using a reduced representation, DD-RAD-seq, I created de novo artificial reference genome and through extensive filtering steps ended up with 174 individuals across 9 sites. I had about 2 million reads per individual and 6,000 SNPs with an average of something like 10x coverage depth. And right away, super deep divergence. Again, these are difficult to see from the back, but that percentage of variance explained on the x-axis for PCE1 is 60%. So initially, I thought something was wildly wrong in this data. It's not. I ran this a whole bunch of times, and there you see the green ellipse, which corresponds to the same color on the map, separating out the Pacific drainages, and the purple ellipse with the Arctic, and a very, very high FST value. But this doesn't really tell us a whole lot other than that these are deeply diverged. I ran another analysis. I'm looking at a maximum likelihood phylogeny using RAXML. This is important because it's not based on allele frequencies, but you can still see this deep divergence. You see the sister species on the bottom in red, that's Lennox, and then you still see the deep divergence between all of the Pacific fish in the upper hand and the Arctic fish in the lower. We also found way lower diversity in the Arctic drainage. This is consistent with what we know about historical patterns in cold water salmonids, and the geological history. Much of the area upstream of Lake Baikal in that purple region was covered in ice sheets for a lot of the times, and those ice sheets came and went. And then this is also consistent with what we know about different Arctic species. So think of the classic example with Atlantic salmon experiencing quite a few bottlenecks and expansions throughout geological time. So this is interesting. We have this Arctic drainage that didn't show any population structure, which we expect, and high levels of outbreeding. So I decided to look at each of those regions separately. The first, you can see this is the Arctic drainage, and this is what I like to just affectionately call a confetti plot. There's not a lot going on here. You really can't tell a lot about what's happening in the Arctic, not a lot of structure. And this makes sense, again, thinking about those really wide home ranges for individuals. So in the Pacific, wildly different results and very, very surprising. So this is looking at the Onan and Amur, the first of the Pacific drainages, that region up in the pink in the left-hand map. And those individuals grouped together. And then the Tagore, that very, very small map that you see inset on the right, the small scale, individuals separated by only a few kilometers in some case, showed really highly structured populations. This was not due to sequencing, bioinformatics errors. I went through everything thinking this might be missing data, and that was not the case. So I decided to look at what was happening in the Tagore River system. So here, just something to keep in mind as we go through, that pink cluster, those upper and lower Onan sites in the Amur River drainage, the very, very large drainage, seem to be somewhat clustered more closely with the Tagore tributary populations. Those black dots are the Tagore main stem, and that Tagore main stem parallels, there are two, and it's a bit of a wetland type of area. So I used a coalescent-based demographic modeling approach to infer the demographic history and try to understand some of these signatures of evolutionary history in the Tagore populations. If you were at William Hemstrom's talk, maybe, bioinformatics talk, it was the last talk of the computational biology, you heard a little bit about the site frequency spectrum. I'm not going to hound into that, but essentially the site frequency spectrum contains signatures of demographic processes like migration or gene flow, divergence times, ancestral population sizes. So if you're looking at this, I'm just going to show 4 different example models. Time is moving up. So at the very bottom, you have an ancestral population in gray that's moving from time 0, that first tick mark on the left, up to time 1, up to present. And so you can feed these models. You essentially estimate the empirical site frequency spectrum, what's in your data. Then you feed the models, you set these parameters. You say, I want you to assume in this case an example that has no migration. You have a divergent split, there's no migration between those populations. You get that, calculate that site frequency spectrum, and then you compare those spectra to the empirical data and find the one that most closely matches your data. So this is a 2-population example, 1 split, no migration. In the next example, we have an initial split with migration, so gene flow initially, followed by a time period of allopatry. Here we have the opposite, so an initial divergence in allopatry followed by secondary contact. And in this example, we just have continuous gene flow between populations. So this is what we found. This is the model that best fit our system here. So on the bottom, we have an ancestral effective population size. We had a larger mainstem population size with first a divergence in allopatry, followed by secondary contact with more individuals migrating and more gene flow from the mainstem populations into the tributaries. Divergence was 28,000 generations ago. When you think about this species, they are not sexually mature until they're 3 to 8 years old. They're not sexually successful until they're 25 or 30 years old. And this is for a species that lives to be at least 80, I would argue 80 to 100 years old. We found individuals that old, and I would imagine that the very large unicorn fish out there are quite a bit older even. So when we're talking about 28,000 generations, this is a long time. This is a long time of divergence. Whoops. These are just the points I already said. Okay, so we have this cryptic divergence in the Tagore. Again, the allopatric phase followed by colonization. This could really match with paleohydrological data that indicates that potentially those ice sheets and stream capture towards the Pacific Ocean caused flooding across the landscape between the Tagore tributary populations and the upper Amur River basin populations, so a little bit more gene flow there. This also is really, really interesting because there are no barriers to movement at all in the Tagore system. There's some differences in chemistry, there's some differences, some other minute differences, but I don't know, is this a runtime issue? Is there something interesting happening here? These are not anadromous species, but maybe there is something. There is an anadromous sister species, but they're a different genera even. So, again, cryptic divergence in the Tagore despite physical barriers. And this is just cultivated a lot of cross-cultural, cross-government, universities, just collaboration. So coming sort of back full circle to thinking about why you do science, this is really, really important working for nomadic people in an area that is often unstudied and underrepresented. So it's just a really great way to be. I highly encourage you to harbor these relationships with other universities, other governments, and cultures. So with that, I have a ton of people to thank. This is just one of the science teams that came out with me. And with that, I'll just take questions. Thank you for coming to a not adaptation talk.
I have a question for you.
Yes.
Just looking at your Arctic versus Pacific, would you argue it's two different species?
Oh, the species, the S word. Knee-jerk reaction, absolutely. Everything about them is different. They are different behaviorally. All of their ecology is different. They look completely different. They act different. I would very much argue that that's the case, especially with such a deep divergence. And that's part of a potential future direction. I'm not a taxonomist, but I would love to be able to do that only because they're taking fish from, for example, the Russian basins, raising them in hatcheries, and they're talking about restocking some of the depleted Mongolian populations, which, to give you an idea, the ones in the Tagore are those ones that are 100 pounds and this big around. The ones in Mongolia are still this big, but they're more like torpedoes. So they would really cause great competition in the system. So, good question, though. Thank you.
Okay, thank you. Hello, everyone. Thank you for coming to my talk. I'm Laura Steinmann. I'm working in the Novikova lab in the Plant Breeding Institute in Cologne. And I want to talk to you today about the role of the environment in the origin and evolution of Arabidopsis lyrata. Our favorite model organism is Arabidopsis lyrata. It's quite unknown, as also it's a close relative to Arabidopsis thaliana, and it is distributed across the northern hemisphere. But it has a huge polar distribution, which makes it an interesting study organism for adaptation. It also is outcrossing, which is an interesting feature for many study organisms, even because it's different than the selfing Arabidopsis thaliana. Okay. This should be a map with a lot of points, which is not the case, unfortunately. So you should see a huge collection of Eurasian Arabidopsis lyrata, which is mainly distributed across the Russian part. It's across the polar circle and also in Europe. But unfortunately, this is not the case here. We see, I want to talk about two different parts of my project, and the first one is the environmental adaptations of Adopsis lyrata. For this, unfortunately, here we see a map, so we selected our samples, which are on a latitudinal axis on the east of Siberia. And in total, we selected 124 individuals of these orange clades, which don't show a lot of population structure. We select latitude as a phenotype because latitude is an approximation for day length and correlated climatic factors like temperature, for example. And then, with this data, we run a genome-wide association studies and look at genes that might be of interest. And we see this following pattern, which is already a great result, because we see a lot of different adaptation genes. So, for example, we see AB80, a member of chlorophyll AB binding protein family, as well as flowering locus genes, FT and FCL3, heat shock-like proteins like SL1, and SUV2, which is responsible for UV censoring in the plant. This all shows that lyrata has an adaptation to latitudinal patterns. And it also shows that also we have a great benefit of using lyrata for GWAS studies, because it is outcrossing. So although we are just having 124 individuals, we can detect enough genetic diversity to detect these candidate genes. And this is really in contrast to Taliana, but is really consistent with the pi calculations, because Taliana has a pi of 0.02, whereas lyrata has 1 magnitude more pi, which just gives us the opportunity to use less individuals and less sequencing. For the second part, I want to talk about the polyploid aspect of lyrata. So what I didn't talk about before is this lyrata is not only diploid, it also exists with tetraploid individuals. And here I want to look at the predisposition of the polyploid formation in lyrata. A short recapitulation, polyploidization is the event of whole genome duplication where an organism has more than 2 sets of chromosomes. And we know that whole genome duplications are not randomly distributed across time. We know that they usually correlate with climatic changes. And for example, when we date the angiosperms back to where the polyploidization event occur, we usually land up in the last couple of ice times, as well as in the Adopsis genomes, which is also shown here. We also know that we see a latitudinal pattern of polyploid frequency increasing towards the poles, which makes it really interesting because there seems to be an environmental factor influencing polyploidy. And also I want to point out here that the Siberian distribution of our Adopsis lyrata matches one of these gray points where it's not really studied until now. This latitudinal pattern could be explained by 2 hypotheses. First, a facilitating condition of polyploid formation, or an adaptive advantage of polyploidy. And today I want to talk about the first aspect. So how does polyploids arise? Polyploids arise through 2 N-gametes, which can arise through 2 scenarios. It could be somatic mutations which occur in one branch and lead to 2x pollen in this branch in regard to the other branches which don't occur whole genome duplication and have still 1x pollen. It can also occur through 2 unreduced gametes in meiosis. What this means is in diploids, if there is normal meiosis going on, it will be reduced to haploid gametes or 1x gametes, whereas when meiosis is not working properly, there is 2x gametes produced. In tetraploids, this would mean that through a normal reduction of meiosis, we have 2x or diploid gametes, whereas when there is no reduction going on in meiosis, there is a 4x gamete. We also already know artificial factors how we can trigger 2N gamete production. So for somatic mutations, we know that we can treat plants with colchicine, triggering whole genome duplications in somatic tissue, as well as we know some Adopsis thaliana studies where it is shown that when you knock out meiotic genes, you can find an enormous amount of unreduced gametes produced. But we want to look at these questions in a natural setting. So we have a collection of live populations of Rhodopsis lyrata which we can grow in the greenhouse, and I here indicated different lineages of these plants. So we have diploids and tetraploids from Europe, as well as tetraploids from Central Siberia, and also diploids and tetraploids, and East Siberian diploids, and I want to compare all of them in a common garden experiment, where I grow them under normal temperature for all of them, and take the flowers and measure the DNA content with flow cytometry. And what we are expecting to see is that we have two kind of peaks, one big peak, the one-end pollen peak, and the second peak with two-end pollen or unreduced pollen. And I did that, and this is how the measurements look like. So this is an exemplary measurement of one plant. You have the first big one-end gamete curve, and the second smaller one, which is here orange, with this two-end gametes. And I now did not do this only on one plant, but on 77 of these plants, and on different accessions. And this is the summary figure which come out of that. I always calculate the percentage of the small peak in regard of the big peak to have a 2n gamete proportion. And here every column respects a population, and each point reflects a sister which we calculated which we grow together in one experiment. And you can see that we see quite some variation of unreduced gametes in all of these populations. In total, the variation ranges between 1.7% of unreduced gametes up to 29%. And this is not only true for diploids, we also see the same pattern in tetraploids, which is here orange. We can also look at the geographical distribution and see already some preliminary correlations that some geographical areas produce more unreduced gametes than others. And in total, this is a first indication of a genetic component involved in 2n pollen production, since we grow all of these plants under the same conditions. And this is one of the next steps which we want to find out which genes are underlying these proportions. But I want to confirm you that these measurements are real, and I highlighted the three most producing unreduced gametes candidates, and I show them here. So we always see this huge diploid peak, but since we have here always a huge unreduced gamete peak, we see this second peak coming up in the back, where we see always a really different of the noise. And what we also want to do now, and this is one of the next steps, is we want to see if we can change these unreduced gamete pattern by changing the environmental conditions where we grow these plants. And the hope is that when we change environmental conditions, we see differences in the unreduced gamete production. So as a summary, I would hope to have shown you that Rhodopsis lyrata has adapted to their latitudinal spread involving genes adapting to UV temperature and for flowering time, for example. I have shown you that Adaptation lyrata is a suitable model system for genome-wide association studies due to their outcrossing behavior, so you don't need to sequence that much individuals. And Adaptation lyrata shows high variation in 2n gamete production in different accessions, which makes it a great study organism for polyploid research. And as well, there is a first suggestion of a genetic component involved in 2n gamete frequency, which could make Adaptation lyrata predisposed to produce polyploids, and this is the reason why we see them in Adaptation lyrata and not in Adaptation thaliana, for example. And with this, I would like to acknowledge my group and other collaborations with where we sampled our plants, and I'm happy to take questions. Yes?
Do you expect the high resulting populations of Adaptation lyrata to have higher or lower amounts of unreduced gametes?
Yes, so this is really interesting. Maybe I can go back there. So we see Adaptation thaliana has two selfing populations, one in North America and one in Siberia, which we detected. And actually, so they are the latest diploids nearly at the orange ones. And they have exactly opposite unreduced gamete productions. So one of them is quite on a low scale of unreduced gamete producing, and the other one, which is the Siberian one, is much higher. This is also a really interesting effect. Yes.
So what type of environmental factor you really want to test to see if there is an effect on production of unreduced gamete?
We mainly want to use temperature, because since we know that ancient times of climatic change usually come with temperature changes, it's the first factor we want to test, but we also think about precipitation.
It's a good chance this is a boring question, but the plots that you showed us in the distribution of the DNA size, they varied in the amount of noise that was happening.
Yes.
Is there anything interesting there?
No, I think so. Pollen, if you make flow cytometry with pollen nuclei, usually you have some, so flow cytometry works on a laser basis, and you have some reflections that are representing this noise. And regarding on your different metabolites in the sample of the plant, it could be quite different of how it actually reflects in the flow cytometer. We are working on reducing that, but it's not always easy. There are no more questions. Thank you for attending my seminar.